and the encouragement during years work and chatting about the science. It's always enjoying talk to you. Let me see how I move the slide. Okay, so today I'm going to talk to you about a topic you are not very familiar with. You know the T-cell therapy, but you probably never heard about the attIL12 T-cell therapy. Hopefully today I can explain to you what is the attIL12 T-cell therapy, what is the implication, why we are interested in create a different category for T-cell therapy. Consider there's already a flood of T-cell therapy available in the market. So by sharing this information, hopefully I can get some talented feedback from you. And also you can learn the general aspect or the arena of the T-cell therapy scope. So for the left panel, you can look at this standard CAR T-cell therapy, which you heard a lot because they are very effective for treating leukemia or B-cell leukemia patient. But the literature or the news didn't really tell you very informatively. Is the CAR T cells effective for B-cell and for some lymphoma, but not effective for solid tumors? You probably ask why. One of the primary reasons all the CAR T cell therapies focuses on killing tumor cell. In other words, everybody or most lab effort to make CAR T cells, how make the CAR T cell healthy, how make the CAR T cell survival long, how make the CAR T cell effectively kill the tumor cell. That's all effort about killing and survival. But our attIL12 T cells, the focus is not really about tumor cell killing, because if I'm focused on killing, there's already CAR T cell. People have done a super job. So instead of compete with the CAR T cell, let's see whether we can find a niche to help each other, synergize each other. So we develop a therapy, so I call it antigen spreading and the stroma disrupting. You probably say, what is hell for antigen spreading and the stroma disrupting if you are not an immunologist? Vice versa for the immunology, if you are not a cancer biologist, you say, what kind of T-cell therapy you talk about? So as the slides go through, you are going to understand. But here, really briefly, want to introduce the CAR T-cell in general. Have a sort of an antibody single chain fragment which can bind in tumor antigen. For this case, the example CD19, because that's effective for B-cell and B-cell tumors. And then there's a transmembrane domain, but really the major things is not only the outside, but also the inside, have 4-1BB and zeta, which I will show you why this is important. But for our interleukin-12 or attIL12, it is similar. We also have a tumor targeted antigen, but it's only 7 amino acid. Only 7 amino acid. Did we try antibody? We have tried one antibody, but I'll talk to you more, but didn't work. So we keep try the 7 amino acid that we discovered can target to tumor. We also have a transmembrane domain, similar to the CAR T cell, have a transmembrane domain. In the middle is interleukin-12. Why we want to make the interleukin-12 into such a structure, I'll show you later why we changed this, because the wild type, in brief, the wild type 12 is a very toxic. In theory, the 12 is as good as a PD-1 blocking antibody, but the truth is the 12 is too toxic to be used in real clinical setting. So why the CAR T cell can kill tumor cells, why our attIL12 T cells have a little tumor cell killing. It's not saying there's no tumor cell killing, but at least in vitro, very few tumor cell killing. If you give it to in vivo, there are some tumor cell killing, but still it's not a major mechanism. But the CAR T cell highly relied on tumor cell killing. Why the CAR T cell can kill tumor cell? Like I said, there's antibody can bind the tumor antigen, but most importantly, this binding is a trigger internal CD3 and ZAP activation, which activate the effector molecule, such as the perforin and the granzymes, interleukin-2, all those things can penetrate the tumor cell and make pores in tumor cell. The granzyme A and B and those things can penetrate into the tumor cell, kill the tumor cell. That's why so effective. That's why the people are so interested in the CAR T cell, because it really gives you a direct visualization. You can kill the tumor cell very actively, make you excited. But this one is, if at least in vitro assay, you can see some tumor cell killing, but it's not nearly as exciting as this one. And you are not going to see a lot of effect because they are really dependent on the antigen spreading, stroma disruption, and followed by tumor cell killing. I'll explain how. So another group of T cell, as the T cell therapy involved now the world is also not only focused on CAR T cell, but a lot of excitement about TCR T cell. TCR T cells is more like a natural occurring CAR T cell. In other words, the T cell actually already build a mechanism. They can use their T cell receptor recognize tumor cells through binding antigen presented by HLA or MHC class 1 molecules. So the T cell already built such a mechanism. But the problem is the TCR T cells, one of the million cells, in other words, it's very rare. So it's very hard for your own body to respond effectively. But if you can artificially isolate this T cell outside of the body, and it can expand ex vivo, then give it to the patient back, of course, can play the same role as the CAR T cell. Actually, it's more effective and more safe because they occurs naturally. And the part of the reason their binding mechanism perfectly matched, and also they have a lot of accessory molecule triggered this CD3 and zeta activation to activate to secrete the pores granzyme to effectively kill tumor cells. So the attIL12 has a little bit of homology functionally, not structurally, but functionally has some homology, share some similarity. Because attIL12, like I said, can induce antigen spreading. What is antigen spreading? Basically, it's induce endogenous T cell expansion. In other words, the host very much has the T cell, even don't have it. So the attIL12 T cell can help dendritic cell to trigger the T cell induction. So this is a difference between the CAR T cell, TCR T cell, which directly kill tumor cell, but this attIL12 T cell can help trigger the TCR T cell activity or induction. So you also probably help from engineers like Ravani's work and others' work about NK cell therapy or gamma delta T cell therapy. Basically, they use NK cell receptors such as NKG2D to interact with NKG2D ligands such as MICA A and B and so on to interact to kill tumor cell. Again, it is a killing mechanism. As long as you engage the granzyme perforin secreted, so make pores to kill tumor cells effectively. You probably also heard about the Fas ligand, Fas TRIL, and TRIL receptor, ADCC, anticentral mediated the tumor cell killing activity. All those are based on direct killing. But the attIL12T, again, is based on the antigen spreading But probably you can also arm the attD12 T cell to the gamma delta T cell to the NK cell. I have not personally done that. We just focus on attD12 invention at this moment to induce the antigen spreading. But all this mechanism is good to overcome the tumor resistance, but also everyone has some issues to overcome. So, and here's the issue. The current T cell, and NK cell can effectively kill tumor cell killing. Why I'm still interested in the attD12, I have shared the structure with you. And one of the reasons for the solid tumor I mentioned is not effective by either NK cell or CAR T cell, T cell, or other type of T cell so far. Yeah, you have seen some positive report, but in general, the solid tumor still is a challenge use any of a T cell or NK cell therapy. And one of the reasons I draw artificial wall. And then this wall means the stroma. For the tumor biologist, very clear what's the stroma. Basically, the structure between the tumor cells, they're fibro, and called cancer, source of the fibroblast, collagen, integrin, extracellular matrix, all the things you can think about. And basically, I simply described it as a wall. And because this wall, the tumor cell and the T cell and T cell don't really have a chance to engage the T cell. In other words, no matter how effective your T cell and T cell are, if they don't penetrate into the tumors, they are not going to have any major impact for solid tumors. Maybe to have an impact for tumors less than 4 or 5 millimeter tumors. But as soon as it become a little bit bigger than 5 millimeter tumor, they have a hard time to penetrate. So that's why I think there's a wall you have to break the wall. So this is artificial, but this is a real mouse tumor staining. And then this is the tumor edge. This is more close to the center. This is the center. For the red color, you can say it's FAP. And the green color, you can say is fibronectin, but you can stain in other things, collagen, integrin, you can stain in all the stroma proteins. But what I want to draw your attention is in the tumor edge is very, very heavy. In other words, the tumor and the surrounding normal tissues clearly have a wall-like structure and separate the two to make it into a very nice tumor environment for tumors to expand, to survival, to escape the immune cell attack. So if you want to have any success, either it's a T cell and NK cell therapy, you have to break this wall and between the tumor and the normal cell to get a penetrate. Even the T cell, NK cell start to penetrate, still inside there's a lot of stroma proteins, antigen, cells, and to actually block the T cell, NK cell, or suppress the T cell, NK cell, or exhaust the T cell, NK cell activity. That's why a lot of people study T cell, NK cell exhaustion. But to me, this is really the focus is on penetration, how we can break this wall. So that's why we developed this attD12T. So the attD12T, I mentioned the tumor target peptides 7 amino acid. So this 7 amino acid is well published in our paper. There's no secret. And so don't neglect this 7 amino acid. This 7 amino acid is so critical, so important. They can do the chore, basically break the wall. And here's the real example I can show you. So if you give the control T cell, infuse the control T cell into the tumor-bearing mice, So you can say the Fab, fibronectin, collagen, all the things I have not stand. I only stand the fibronectin. You can say the heavy blocking uh, between the normal tissue and the tumors. But if you infuse the same tumor-bearing mice with ATT12 armed T cell, in other words, ATT12, this contract medical virus, and transfected into the T cell, you can infuse this T cell into the tumor-bearing mice, and you can do this side-by-side staining. And here you can see the red color and the green. The green against fibronectin, the red is the Fab. You are going to see very little left over for Fab and fibronectin. And if you look at the more towards the center area, even less. So that's why I say the ATT12 T cell can really help disrupt the stroma. So that is really microscope structure. So how about to look at the entire tumor section and give you a little bit for globally. So this is again the control tumor bearing mice and infused with control T cells. So you can look at the tumor edges, and between the tumor edge and the normal tissue, you are not going to say. But what you can see is this red color. So this red color basically is collagen staining. You can see the entire tumor either is the edge or in the center. The center is less intensely stained, but still there's a collagen. The collagen is all over the place. But what happens if you give a ATT12 T cell infusion? So almost like you wash off the collagen. In other words, it's almost you put the tumor in a laundry machine after washing, you got a clean tumor section. It's almost like that. But the truth is really the side-by-side collagen staining, but you don't really see much of a collagen left over in the tumor. So that's another evidence to tell you this ATT12 T cell looks like the structure. It's very simple, but it can disrupt the stroma. Probably say, well, how this happened, you never. So the truth is we don't know exactly. We just slowly start to understand the mechanism. And here's and I give you a little bit more information about the change and or comparison between the different type of interleukin-12, because we are trying to understand that this is a interleukin-12 effect, or this is a really ATT12 effect, or it's a membrane anchored 12 effect. Because remember, interleukin-12 is a secreted protein. It's a secreted outside of tumor cell. It's a free protein in theory can travel anywhere, but we artificially anchored on top of a T cell membrane, and also bear with the tumor targeted the semi-amino peptide. That's why we need to understand which molecule give us the function to disrupt the stroma. It is the interleukin-12, or it's the tumor targeted peptide, or it's the membrane anchored transmembrane domain. So, and here's we infuse the control TIL. You can see it's much better in terms of collagen staining. In other words, even the TIL actually can reduce the collagen staining, but compared to the ATT12 TIL, still is way much better with the ATT12 modification. But if we have an anchored interleukin-12, in other words, we remove the tumor targeted the semi-amino acid by simply remove semi-amino acid, you can see a lot of collagen is re-emerged. How about the wild type 12? In other words, this 12 can secrete outside of the T cell we infuse to the tumor bearing mice. And we got the same collagen as we can see in the membrane anchored 12. How about the tumor targeted 12 without the membrane anchorage? Yes, we got the same effect as the ATT12. In other words, the tumor targeted 12 12. In other words, the same amino acid, the tumor targeted peptide is very, very crucial to trigger this impact. Then you probably ask me, why don't I use the tumor target 12? Why I use the anchored? And I'll show you later, because the toxicity. Any wild type 12 after secretion outside of the T cells, very poison, can kill patient. And the tumor target 12 can reduce the toxicity, but still there's toxicity. As soon as you anchor to the surface T cell, there's no toxicity. I'll show you later. So this is about the collagen. How about the fibronectin? The fibronectin showed a similar trend. So I'm not going to repeat myself. So this is basically a summary slides, kind of and remind you again to what is the difference between the current T cell versus the attIL12 T cell therapy. At least I show you the difference in terms of the stroma difference, but I did not show you the T cell infiltration attIL12 T cell because we break the stroma. Of course, this time the green color staining T cell infiltration, but I believe I have more slides to show you why the stroma disruption matters. So even though people didn't really give us much of credit, but the people slowly recognize the significance of anchor interleukin 12 to cell membrane. You can look at the CAR generation. You heard of first generation, second generation. Now most clinical trial or clinical approved product is the third generation. But when you look at the fourth generation, fifth generation, and then they actually incorporate the membrane anchored interleukin 12 in different ways into their construct. What I believe is sooner or later, people either give us credit or not, they are going to, one way or other, incorporate the interleukin 12 in the membrane, either develop a T cell therapy, NK cell therapy, CAR T cell therapy, NKT cell therapy, or gamma delta T cell therapy, because they are really friends. They are not competitors, because ones can give the advantage of antigen spreading induction. The other one is really cell killing. But if they are kindly including our tumor targeted component, they probably found more application. What I mean is the stroma disruption. So I talk about this attIL12 T cell. I brag about this 7 amino acid, the tumor targeted peptide. You are going to ask me what I'm targeting, what the 7 amino acid talk about. And here introduce another concept, is the cell surface vimentin. I call the cell surface vimentin because the vimentin is present intracellularly in the normal mesenchymal cell, such as the PBMCs, the white cell. For the tumor biologist, it's well known as present intracellularly inside the tumors, regardless of mesenchymal tumor, epithelial tumors. As long as the malignant tumor or highly malignant tumors, there's tons of vimentin. Without any education, everybody knows. And also play a very crucial, important role for metastasis for many things. But the people in general don't know. So cell surface of vimentin is also present on the highly metastatic tumor cells. And basically, we are pioneer to this discovery. We found regardless of tumor types, either the sarcoma, which I'm talk about today, is most osteosarcoma example, or epithelial tumor cells, any type of tumor cells and their cell surface vimentin. Maybe you are lucky you found a few cell lines, cell surface vimentin negative, you can serve as a control. But you probably ask me why cell surface vimentin is widespread, why people always look at intracellular vimentin, they don't look at the extracellular cell surface vimentin. And that's because the people generally think about the vimentin is functionally localized or present in intratumorally. That's how they function. They don't know there's a different vimentin. Actually, vimentin can present in nuclei in the cell surface, it can secrete it to the blood, can present it to the cell surface of vimentin. As time goes on, people are going to slowly recognize some of the metastatic function probably is induced by cell surface of vimentin. At least we published one paper how cell surface of vimentin can really maximize the metastasis. Can basically the bottom line can beat any known stem cell markers, because stem cells known as a tumor cell, tumor stem cells known as sort of a metastasis cell. So we can use cell surface vimentin positive cell can compete with any known tumor stem cell biomarker to compare which one can metastasis faster, at least in tumor, liver tumors, so cell surface vimentin stem cells really, but that's another story. But here, what I want to emphasize, this semi-amino acid is really targeted the cell surface vimentin, and here I give you the breast cancer, bladder cancer, liver tumor, and osteosarcoma, neuroblastoma, colon tumor. We collaborated with many people. I'm always so afraid to mention any because there are so many people, especially clinicians, clinical teams collaborate with me. So make us possible to make this observation and a discovery. And also I'm very proud that this is occurs in MD Anderson and in other institutions. We don't really have an opportunity to can collaborate with so many clinical teams to do the chore. So then this is initially how I discovered the tumor targeted the 12. For the time of the sake, I'm not going to repeat my published work. I just want to draw your attention. This is a semi-amino acid. It's no secret. It's called a Van Tenest. So you are welcome to try in your gene construct. But what I have to share with you, the people always ask me why you focus on interleukin-12. To be very honest, I have inserted this peptide to many different genes. But so far, it's really fantastic when I insert right before the stop codon to the interleukin-12, works so beautifully and it really help. But if I work in some of other genes, I don't really see much of a benefit. But in fact, we have a terminate of those, and some actually have an opposite effect, but it really work with tumor target 12 very, very effectively. So now I give you I can give you one piece of data about the tumor antigen. I talk about enough stroma, how the attIL12 T cell can disrupt the stroma. And also I told you the second function is the attIL12 T cell can induce the antigen spreading. What is the antigen spreading? I already told you earlier, basically it's induced of endogenous TCR T cell expansion. And here's actually give you a real piece of evidence to support this attIL12 T cell can induce antigen spreading. But for the antigen spreading, obviously you cannot use immune deficient mice. You have to use immune competent mice. So this attIL12 T cell here is talk about the mouse attIL12 T cells. For the mouse attIL12 T cell, we use the OT1 T cell. For the people who are not immunologists, probably get irritated and say, what is the OT1 T cell? You don't have to worry about it. This is just a tumor-specific T cell for the mouse, and they recognize all expression tumor cells. So you can just consider as a TIL or tumor-specific T cell or TCR T cells. So because the tumor-specific T cell, if you give it to the tumor-bearing mice, you would expect that there's some effect. So this is a B16 melanoma tumor cell expresses OVA. And then if you give this OT1, no attIL12, just OT1, even though this OT1 recognizes B16 expresses OVA, but still the tumor can express rapidly because any immunotherapy, if you give the tumor a little bit late, the tumors are going to grow because they can escape the immune mechanism. Why? Because the solid tumor develops stroma, they can avoid the engagement between the T cell and the tumor cells. Really say, which one can win the battle. And sorry, the red is the control tumor. But if how about I give the OT1 tumor cell, yes, can reduce the tumor growth significantly, but still tumor grow rapidly. So the orange color tumor still. How about the blue? Blue is the wild type interleukin 12 and a very toxic 12 produce OT1 cell. And it can do a better job, but the problem is toxicity. How about our attIL12? attIL12, the green, attIL12 modified OT1 cell. The tumors shrink. So that's nice. Even the strength, but some of them might still grow small tumor. But regardless they grow tumor or not, I just surgically remove all the tumor. And just pretending I'm a surgeon, but I'm mouth surgeon, just remove all the tumor. Of course, it's not me. I'm not as nearly as good as my lab members. My lab scientists done the surgery, remove the tumor, all the mice tumor free. So now they have a zero tumor. Now they have a zero tumor. I just artificially reimplant the tumor, basically grow a tumor B16F10. So I emphasize B16F10. I did not mention OVA, meaning so OVA negative B16F10. Why this is important? Remember, this OT1 cell recognize OVA expression tumor cell. Now I re-challenge the surgical remove the tumor-free mice, and with B16F10, no OVA. So if I give OT1, and useless because they don't recognize. But I do basically challenge this free mice with the wild type interleukin 12 OT1 in the blue, the control T cell. Sorry, I didn't challenge T cell, but I just give the over negative B16F10 tumors. Then I monitor tumor growth. So what you can see is the attIL12 OT1 treated mice in before the surgery, they still no tumor relapse, no tumor relapse. They are tumor free. But the wild type challenge the things, they develop tumors, not aggressive, but they develop control. Of course, they develop tumors. So why this is tumor free? This is called antigen spreading. What is all the antigen spreading? Again, it's attIL12 T cell. In this case, attIL12 OT1 induce the TCR T cell that does not recognize the OVA, but recognize the other. That's why even you give a OVA negative B16F10 tumor cell, they still don't grow tumors. That's why they call it antigen spreading. That means they can recognize beyond the OVA, even beyond the OVA, meaning OVA negative tumor antigen. That's okay. They can deal with those tumors. That's why I spend a little bit of time because I expect that some of them not immunologists. So those are immune competent mice, but even in immune deficient mice, regards to colon tumors, osteosarcoma tumor or lung tumor, and you can see the bottom one, you can also see some impact. And the part of the reason is by disruption of stroma tissue, and the part of the reason by induce some tumor killing effect by induce NKG2D ligand in tumor cells and NKG2D receptor on T cell. So use this mechanism and induce some tumor cell killing. That's why even in immune deficient mice, you can see some impact. But one important feature, which eventually I can convince myself more this attIL12 T cell into a clinical trial, it is the safety feature because my lab has been working on this attIL12 T cell for 25 years. I started my lab 25 years ago when that times interleukin-12 is as good as current days talk about anti-PD-1 antibody. It's really a cool molecule, but the only frustrated things to clinicians toxicity. So as a research investigator, when I started my lab, my whole goal is can I really make this interleukin-12 toxic free? And now I can comfortably to make this announcement, at least in mice, attIL12 T cell, it is absolutely safe in mice. And here's a few examples. One of the mechanisms for interleukin-12 induced toxicity is by induced TNF-alpha and interferon gamma and interleukin-6, all those known toxic cytokine release syndrome cytokines. But if you use attIL12 modified T cell, attIL12 T cell, and you can see the last one, say, even compared to the control, no interleukin-6, no GM-CSF. Of course, controls don't have GM-CSF. The TNF-alpha, say, compare this is a wild type interleukin-12 T, have a lot of TNF-alpha, but attIL12 T, none, just like a control. The gammas here use the scales, so you don't really see much difference, but also actually significant. So this is a very critical slide to use, a very simple slide, but this is one of the critical slides to convince FDA to prove our IND. So of course, I don't rely on those single slides. We've done a lot of histology to show you how this attIL12 T cells, instead of, if you give the control T cell, even the CAR T cell, you heard about some of the CAR T cell, control T cell, after infusion in the patient, they stuck in the lung and storing in the lung. So the lung has to, that causes lung toxicity. So if you give the wild type 12 T cell or membrane anchored 12 T cell and also stuck in lung, but if you give the attIL12 modified T cell, there's nothing in the lung. So this is another crucial data to convince the FDA and to show the safety feature of this attIL12 T cell to allow us to launch the clinical trial. So, and also FDA loved these slides. So the left part is the infusion control T cells red, that's a collagen. So in the right side, so you can see the blue color, you don't see, well, there's some red, but you don't really see near as bad as the left is infusion attIL12 T cell. So in the osteosarcoma, at least in the osteosarcoma, I don't really see much of collagen, but you see a lot of green color in the center of the tumor. The green color CD3 staining, that's a T cell staining. But in the control, yeah, you see maybe one few dots, but not nearly as many as in the right panel after infusion attIL12 T cell. This is another part to get the FDA excited. Now finally, and also remember, I told you this is the highly malignant metastatic tumor cell surface has cell surface vomiting expression. So, and to know in the future, not now, not for the phase one trial, for the future trials, we can always just draw blood to look at the circulated tumor cell, which tumor or whether the patient we selected, it is a cell surface vomiting quality, almost 100 patient tumor, a cell surface vomiting quality. But the problem is for the primary tumor patient, maybe only 5% of the tumor a cell surface vomiting positive, 95% is negative. But for metastatic tumor, maybe it's opposite, maybe it's 50 to 17 to 90% cell surface vomiting positive, but 50 to 10% is negative. But regardless, at least we can do a blood testing. So finally, I want to share the good news. This is FDA has approved our IND with all the good work from our GMP team, our regulatory team, our IND office, and my lab, and everyone who helped me in one way or other. We finally launched the first attIL12 T cell trial. Actually, next week it's going to be exciting because it's our first planned infusion of a first patient. So this one is funded by American Cancer Society and led by Andy Livingston, who will serve as a PI. But really, our pediatric team like Dristy, Dr. Dristy Ragnall contribute a lot. Whole transplant team contribute a lot. So now I'm closely work with the GMP IND team to file the second IND attIL12 TIL. So I'll keep you updated. So there's a lot of challenge regardless of our current attIL12 T cell therapy or the CAR T cell therapy, TCR T cell therapy. There's a lot of work to do. And so I guess I'm going to, there's I probably share this slide. There's one challenge we could do is, like I said, our attIL12 T cell is not a competitor of a CAR T cell therapy, not a competitor of a TCR T cell therapy, not a competitor of NK cell or gamma delta T cell therapy or anybody's competitor. We are a friend of yours. Here's one example. This is a B7H3 CAR T cell example. And this is one of the osteosarcoma. It's from Richard Gordick's lab, and he provided tons of osteosarcoma PDX model to us. So you can look at the this circle dots, which we infuse the tumor berry mice with the B7H3 CAR T cells. And even though you saw the tumor's progression, the truth is actually slowed down quite a bit compared to the control tumor. But when you combine, but the problem is tumor still progression. But when you combine with our attIL12 and this B7H3 CAR T cell, you can see the true tumor regression. And then more impressively, you can look at the tumor volume, the start volume. So this is talk about 300 cubical millimeter tumors. It's talk about the start point is 1 centimeter in diameter. Most people treated the tumors, you know better than I do. Probably you start at 5 to 6 or 7 or 8 millimeter tumor diameter. This talk about 1 centimeter tumor diameter tumors. We still able to regress the tumor. This is called OS9. I'm sorry. So it's called OS9 in the tumor model. So that's why what I'm saying is this is your friend. So you are welcome to collaborate. I'm not a compete with anyone. So this is really part of our lab story I like to share because I started my lab interleukin-12 therapy all the way to the clinical trial launching. And hopefully in the future, I can share more exciting data about the clinical trial results. But that's really part of my lab story. We also study other sort of immunotherapy. One major story is about FGL2 target the T cell therapy. It's not about the target T cell therapy. Again, it's about the concept. The concept, like people's killing tumor cell. What we are trying to do is how we can induce a resident memory T cell in the brain, make the T cell living in the brain forever to reject the tumor relapse. So that's another exciting story my lab is developing. So there's many collaborators. Even though I list the collaborators, I'm always cowardly to say any of the names because the truth is there's too many to say. And also this is all the slides. As time progress, I always have a new collaborators. I'll stop here, say whether you have any questions I can respond.
So congratulations, Shulin. This is really wonderful. It's really wonderful talk. And in fact, it's basic science that you was able, and of course, your colleagues, to bring to clinical trials. This is the essence of great work at MD Anderson. Let's start with a question. Great talk, great question. So it is a question here from Dr. Sam Hanash.